So in this particular delivery, I'll be only giving you an overview of the genesis and the history of bioinformatics, the areas covered under bioinformatics today, and what are the important applications, giving certain important examples. So I hope that the subsequent lectures to be delivered by the eminent speakers from tomorrow onwards will give you details about the different applications of bioinformatics in different branches of life sciences. But today I shall just give you an insight into the different areas and applications in a nutshell, giving an overview on the subject. So let us first discuss what is bioinformatics. As you might be knowing that this particular term itself is new to the life science researchers, because the term was coined only in the year 1970 by a scientist called Pauline Hogwag. But the context in which he used the term is not applicable today, because he used the term to refer to the study of information processes in biotic systems. But the modern definition of bioinformatics was first introduced by Dr. Hua Lim in 1987 after the advent of the omics era. As we know that after the advent of the sequencing technology, when people started working on sequencing of genes and genome, numerous biological data were made available out of the genomic projects. The quantum of data was so huge that it was not possible to store, analyze, and interpret the complex data by manual process. So people started using computers for not only storage, but also for analysis and interpretation, so that the complex and heterogeneous biological data can be analyzed appropriately, and all biological functions can be correlated to the intricate interactions among the biomolecules occurring within the biological system. The modern definition of bioinformatics is still evolving, because with the advent of the different areas in bioinformatics, many new applications have emerged in the last few years, and therefore it is now considered as an interface between biological sciences and material sciences. This is a marriage between biological and material sciences, and many new areas are being explored wherein principles and procedures which are conventionally being applied in different biological sciences like chemistry, physics, information technology, computer science, engineering, mathematics, statistics, etc., are increasingly being applied in terms of data analysis and interpretation in biology. So therefore, with the increasing use of applications from material science, many new areas are being added to bioinformatics, and the definition of bioinformatics also is evolving. So that's why I have put here in this slide some of the newer concepts or definitions in bioinformatics. The last one, that is, bioinformatics is an integration of mathematical, statistical, and computer methods to analyze biological, biochemical, and biophysical data. It's considered as one of the near complete definition of bioinformatics today. As I already said, that bioinformatics now has become a key component of all branches of life sciences, and it's a conglomeration of our knowledge and skills of both material science and biological sciences. That's why it now takes the center stage among the different branches of material and biological sciences and are applied in wide areas in life sciences. All our understanding of the intricacies of the biological system and its regulation centered around the central dogma in molecular biology. That means the conversion of the genetic code hidden within the nuclear material of a living cell in the form of DNA or RNA in case of viruses, and how it is converted by two steps of translation and transcription and translation to its product. Although at the beginning it was thought that there is a single gene for every product of a gene, that is protein. So one gene, one protein concept was widely accepted, but this does not hold good today because now it is known that gene expression does not lead to production of protein alone, but there are many other molecules like transfer RNA or ribosomal RNA, which are also encoded by. Similarly, a single gene may also give rise to a number of variant protein molecules because of a phenomenon which is called alternate splicing mechanism. I'll not go into detail of those mechanisms which are pertinent to the basic molecular biology, but let us know a little bit about the basics of molecular biology so that we can understand the basic phenomena that we are going to explain in terms of bioinformatics in the later part of this lecture. As we know, that the genetic code in DNA is stored in the form of triplet codons, three nucleotides arranged sequentially in a segment of DNA identified as gene, codes for a single amino acid. Now we have got 20 amino acids, and all these amino acids are coded by triplet codons. A codon consists of three nucleotides. Now we have got four nucleotides, therefore, the four nucleotides can be arranged in groups of three in 4C3 ways. If we expand 4C3 mathematically, it becomes 64. Out of the 64 variants of codons, 3 codons are considered as stop codons and they do not code for any amino acid. Except these 3 stop codons, all other triplets codons code for amino acid. As you all know the basics of molecular biology, that AUG codon in the messenger RNA codes for methionine. And this is also called as a start codon or initiation codon. And that is why in any functional protein, methionine is the first amino acid residue. As 20 amino acids are coded by 61 different triplet codons, therefore, there are many amino acids which can be coded by more than 2 codons. Even there are amino acids which are coded by 4 or 6 codons. So therefore, the codon utilization or usage of codon by an organism for production of protein is also quite variable according to species. So we say that there is a codon usage bias among the organisms. If the codon usage pattern can be studied in the species, particularly in terms of the host-agent relationship, if we want to find out the relationship between the host and the agent like parasites, like bacteria, viruses, or other parasites, then if there is greater compatibility of usage of different codons for different amino acids, there is a greater possibility that biocompatibility between the agent and the host can be found. So, for example, it has been established beyond doubt that among the domestic animals, the SARS-CoV-2 virus, the virus responsible for the current pandemic, shows greater affinity towards dogs among the domestic animals. That means there's a chance of infection in dogs in higher rate than that of any other domestic animal, because it has been established that the codon usage pattern in human is highly correlated to the virus, and the same way the codon usage pattern of dog is also highly correlated to the codon usage pattern in the virus. So there is a greater susceptibility of dog compared to other domestic animals to infection by the SARS-CoV-2 virus. So this is just similar one example I'm giving you. So therefore, in terms of use of specific codon, if we study, then codon usage pattern can also give us definite clue regarding the host-agent relationship. And also, there are many other biological functional activities which are also governed by the usage pattern of codons. So this is one of the areas now in life sciences which give us very important clue in epidemiology as well as in identification of strain and species variation. And this can be studied in greater detail by use of certain tools and softwares which have been developed by researchers for studying the codon usage pattern. Here I'm listing some of the online and offline resources which are available, including some databases as well as the software tools which are commonly used for codon usage pattern analysis. I'll be glad to share my slides for the benefit of the participants. I think you can also individually log into those databases as well as the online resources so that you can get a first-hand idea about how these softwares and resource portals work. I'll not go into detail about that today. Now, one unique feature among all forms of life is the universality of the codon or the coding pattern. The genetic code is nearly universal across different taxa, starting from prokaryotes to higher animals and human beings. The coding system adopted by all life forms are almost universal, bearing very few exceptions. Almost in 99% species, the genetic code is universally almost same. That is why it has given us ample opportunity to manipulate the genetic code and transfer the genetic code and genetic element from one species to the other across different taxa. Therefore, now genes can be transferred from prokaryotes to eukaryotes to prokaryotes, and vice versa, from bacteria to human, human to bacteria, bacteria to viruses, viruses to bacteria, and across different taxa of living organisms. So here, this is one of the pioneering work in genetic research when the gene responsible for the fluorescence that we see in case of firefly at night. So the gene responsible for the pigment that is responsible for fluorescence have been introduced into tobacco plant so that the tobacco plant expresses it in its stem and the root, and therefore tobacco plant can also flourish during night. So this is a typical example of transgenesis of genes or DNA from one species to the other. Now, although the genetic code carries the information in a species for its species characteristics as well as the characteristics of the taxonomic group, but it is found that although the genetic element or the total genome of an organism may be same at different times, the morphology, the behavioral properties, as well as the functional properties of the organisms may differ. A typical example is the 4 stages of the life cycle of an insect. We know that there are 4 stages of an insect life cycle: the egg, larva, pupa, and adult. These are the 2 stages of the life cycle of the same insect. This is the larval stage, and the right hand side is the adult stage of the same insect. But except for the color, there is no any match between the 2 stages of the same insect. But if we go deeper into the genome of the organism, if we extract the genomic DNA from these 2 stages of the same insect, we will find that the genome sequences are 100% identical. There is no any change in the genomic structure. All genes and their nucleotide sequences are 100% identical. But empirically or phenotypically, they are totally different. There is a heaven and hell difference. So this is giving us enough indication that genome cannot explain all the variance or the diversity that you see in the phenotype. Not to speak of phenotypic variation between taxa, but also within the taxa, there are a lot of variations, as this typical example shows. So therefore, beyond genome, we have to study the variation in terms of other biomolecules. So therefore, as I already referred to the central dogma in molecular biology, as you know, that the genetic code is converted to protein or its product by a two-way process, that is by transcription and translation. In initial stage, the genomic code is converted to the code that is given in the form of the nucleic acid sequence or messenger RNA. So messenger RNA sequence is synthesized as per the template of the DNA sequence of the gene. So the first step in the expression of a gene is conversion of the genetic code to messenger RNA, or what we refer to as transcript. And therefore, if we study the transcript, then we can understand the genetic code being transcribed or being converted to the code of messenger RNA. In a particular cell, if we can study all the transcript at a time, suppose there are 700 genes in the organism, in a particular condition, the cell may express, suppose, 500 genes altogether. If we can study all the 500 transcripts at a time, and we compare the transcript sequence to the genetic code, then we can easily determine what are the genes which are under the process of expression. So to understand the gene expression, the first and initial step is the study of the transcript or transcriptome as a whole, all transcripts together. So that is the first stage of expression, and analysis can be done by studying the genetic sequence or the nucleotide sequence of the transcriptome. And when it is further translated into the protein, then the protein will be having amino acid sequence as per the sequence of codons listed in the messenger RNA. And therefore, protein sequence will be directly regulated by the messenger RNA sequences. But proteins are the final product of gene expression. So, as I already said, that all genes are not being expressed at a given point of time. Therefore, if we study the proteins, or as a whole, all the proteins synthesized at a particular point of time, then we can find that all genes are not converted to its product at the same time. So that's why we have to extend our study to different levels of omics technology today, starting from genes and genomics, through transcriptomics, study of transcriptome, and the study of proteomics, study of the proteome, all proteins synthesized at a given point of time. We have now tools for experimental work relating to the expression of genes at different levels, at the genomic level, at the transcriptomic level, as well as in the proteomic level. I'll not go into detail about the methods and their applications because our concern today is the bioinformatics and their applications, not the molecular biology techniques. But there are molecular biology techniques which are available to experimentally work on these lines. And finally, when the genes are converted to the product proteome or proteins, the proteins also do not act standing alone. They do not act on a single protein as a whole. Normally, they work in a network of interactions where hundreds or thousands of proteins may be involved. So proteins and other biomolecules and small molecules present in the living body or living cell will interact with each other in a very systematic manner, in a regulated way under the influence of different enzymes. And finally, they will develop a network-like activities in the form of metabolic pathways. So the pathways of interaction between proteins and other molecules can also give rise to different kinds of byproducts or intermediate products, as well as the final products of metabolism. So the final impact on the phenotype or the empirical effect on the phenotype will be the result of the intimate interactions between the molecules in the form of different metabolic pathways. So these pathway of interactions can also studied separately in a kind of omics background that is called metabolomics. And finally, the empirical structure and the biological function that we observe ourselves as the empirical methods are being applied. Say, for example, morphological measurements, behavioral patterns, the different functional biochemical activities. So those are the final result of interaction between molecules. But we know the conventional biology, which we were teaching and learning till the advent of the omics technology, we are mostly confined to our understanding based on the empirical observations on the phenotypes of the organisms. So phenotypic characteristics were the basis of our understanding of the conventional biology. But today, we have to extend it further to the different levels of omics technology by going into depth up to the level of genes and genome. That is why today in the omics era, we have got different levels of learning in life sciences, starting from genomics, transcriptomics, proteomics, metabolomics, and phenomics. That is the conventional biology. And therefore, it is very essential to go deeper into the molecular level, or if possible to the atomic level, to understand the structure and interaction between biomolecules which give rise to final phenotypic characteristics. So in the process of understanding the life processes at different levels of omics technology, bioinformatics play a very, very vital role. So without the tools and techniques of bioinformatics, now it is not possible to understand the intricacies and the complex mechanism involved in the molecular interactions or atomic interactions that finally give rise to the phenotypic characters. And therefore, bioinformatics play a very vital role today in life science. So any life science researcher cannot avoid learning at least the usage of tools and techniques of bioinformatics. Of course, we may not, all of us, be developers of bioinformatics tools and techniques. We may not be the real hardcore bioinformaticians, but we must have to be aware about the applications and usage of the common tools and techniques in bioinformatics so that we can succeed in our research in molecular biology, and any branch of biological sciences. Now, if I ask you, how much similarity do you find between these two phases? I think it will be a very ridiculous question because I'm trying to compare Miss Universe, sorry, Miss World with a chimpanzee. So, for empirically, if we see the beauty of these two faces, there is no comparison at all. There is a heaven and hell difference. I think I'll be killed by the beauty queen because I'm trying to compare her beauty with a chimpanzee. But for a biologist, intrinsically, at the level of genome and genomic sequence, these two individuals are having 98.7% similarity. Sorry. I think I need not emphasize on the fact that the knowledge that human beings have evolved from certain humanoid apes was well known to us since 1940s. But it was not clearly known which particular humanoid ape was our immediate ancestor, because there are four varieties of humanoid apes. They are chimpanzee, gibbons, orangutan, and gorilla. But after the advent of the genomic sequencing technology, when the genomic sequences, whole genome sequences of all these four humanoid apes were made available, now it has been found that the genetic makeup of chimpanzee is almost similar to human being. There is a 98.7% similarity in the genetic makeup in terms of the nucleotide sequences of the genome. And compared to other forms of the humanoid apes, it is the closest relative to human being. That is why now it is already established beyond doubt that chimpanzees were our immediate ancestors. So we should not ridicule them, we should not laugh at them, because they were our forefathers. This paper was published in Nature a few years back. Now, coming to the different emerging areas of bioinformatics, there are a lot many different areas coming up in the recent past under bioinformatics. Initially, bioinformatics was confined to two areas, that is genomics and proteomics, as I have explained. There are two aspects in both, one structural aspect and the other is functional aspect. Structural genomics and functional genomics means what is the nucleotide sequence of the gene or genome that covers the structural genomics, and how to correlate the nucleotide sequences of genes and genome to the biological activity or the biological property that is included in the functional genomics. That is the gene expression analysis or mutational analysis. Similarly, in proteomics also there are two aspects. One is structure and the other is function. But with the advent of these two omics technologies, the bioinformatic approaches have been employed in a number of different areas in biology today. And some of the very, very important emerging areas are drug designing, computational evolutionary biology, biodiversity measurement, and modeling biological system. It has come to such a time that now, after 2010, human beings have succeeded in not only dissipating or understanding the molecular structure to the extent of the complete genome sequence of thousands of different species of organisms and correlating them with the biological function. But at the same time, they have also reached such a point that now we are able to manipulate not at the gene level itself, but also at the genomic level. And the systemic biology and synthetic biology has come to a great extent now. And in 2010, a group of scientists led by one J. Craig Venter from California, he for the first time introduced a synthetic genome, which was synthesized by chemical process, into a nuclearless or enucleated bacterium, E. coli, and he has converted the bacterium according to the choice. First of all, he implemented this program in a small bacteria called Mycoplasma, which is a cell-wall-less form of bacteria, and he used a synthetic genome of Microbacterium capricolum, having only 167 genes, essential genes, and incorporated it into another species of Mycoplasma called Mycoplasma mycoides, which was enucleated, means devoid of the nucleus. And then, after incorporation of the synthetic nucleus of Mycoplasma capricolum, when it introduced into the cell membrane associated with the Mycoplasma mycoides, the organism was converted subsequently to Mycoplasma capricolum. A change of species also subsequently occurred. And to the utter surprise of everyone, the bacteria not only survived, but it multiplied with the synthetic genome. So from that, a different branch has now come up in biology that is called synthetic biology or synthetic genomics more particularly. So now Dr. Craig Venter has already, he was establishing one company called Synthetic Genomics, and they are producing different microorganisms with synthetic genome, which are capable of producing the desired pharmaceutical products. Not go into details about the applications, but I'll cite few examples of the areas in bioinformatics and which are applied in certain emerging areas in biology today. So one of the most important area in bioinformatics, which was the starting point in bioinformatics, was the construction of the biological databases. So creation of biological databases was the first initiative taken by the people who ventured into application of computer in life sciences. As I already said, from genomic and proteomic experiment, when enormous data is made available, it was become compulsory, or it was a compelling situation for the biologists to use computer to store, analyze, and interpret, as well as to disseminate the information among the scientists. Now, there are thousands of online and offline databases available across the globe, which provide information about both structure and function of genes and genome, as well as proteins and proteome, and as well as many other features which are important for the biological scientists. So these are some of the types of databases which are available for the biological researchers. On principle, today, a biological database has to be an online database shareable across the internet through the global community free of cost. If it is not shared across the globe through the network, then it is not considered as a biological resource for the researchers. So now we have got many databases which store primary sequence data, that is your mostly the DNA sequence data, both of genes and genome, or segmental nucleotide sequences, then protein sequence databases, protein classification databases, pathway databases, microarray databases, two-dimensional polyacrylamide electrophoresis databases, public literature search databases, and patent search databases. These are some of the important kinds, but of course, there are many different kinds of, so to say, classification of databases like primary database, secondary database, generalized database, specialized database, archival database, etc. Three important databases across the globe, which are hosted by three organizations located in three different subcontinents of the globe, are integrated to each other. And they are important resources for all biologists. They are EMBL, GenBank, and DDBJ. GenBank is at the center point because it stores the highest number of data, which are important resources for the biologists across the globe. It is hosted by the government of US in National Center for Biotechnology Information. EMBL, or European Molecular Biology Laboratory, it is hosted by European Bioinformatics Institute in Europe, in England. DDBJ is DNA Data Bank of Japan. This is hosted in Asia, in Japan. All of them are integrated to each other, and there is a intimate connection among them. And therefore, if you enter to one of them, you can get access to the data across others. So they usually cross-talk among themselves so that you can acquire information or access information from each other. Now, of course, EMBL is renamed recently as ENA, European Nucleotide Archive. Most of the European scientists would like to deposit their resources or the data to EMBL, but from US and allied countries, as well as from India and other third world countries, mostly people prefer to store the data or deposit the data to Zenbank. Now, I'll cite a few examples very briefly how bioinformatics tools and techniques can help us in understanding biology. One big problem for the biologist is to identify the genes in a genome sequence. I'll not go into detail about the structure of the genome of human nucleic acid, but you might be knowing that the total length of DNA in the human genome is around 3,300 megabase. 1 megabase is equal to 1,000 kilobase. 1 kilobase is equal to 1,000 base. Each of the base represent one nucleotide. So you can very easily count how many nucleotides should be present in one particular cell of a human being. So the genomic code in human being is too long. There is around 3,300 million bases. Now, although it is too long, the entire length of the DNA do not code for the products, that is your proteins. It has been found that only about 2 to 3% of the entire genomic sequence constitute the genes which code for the proteins. So rest 97 to 98% can be called as so-called junk sequence because they do not code for any product. But it is found that although they were initially thought to be junk, but they are not really junk. They are required not only to maintain the structural integrity of the nuclear material, but at the same time, some of the components of the so-called junk DNA or non-coding part of the sequence also are important for regulatory role to be played. Means there are certain sequences which are called regulatory sequences which control the expression of the gene. Either they allow gene expression at a regular rate, same rate all the time, or maybe enhance, they may enhance at some time, or they may retard or suppress the gene expression, maybe totally inhibit the gene expression in some cases. So these regulatory elements are very, very important. Similarly, in the so-called junk non-coding sequences, the genomic sequence also consists of many signatures of the species or the taxonomic group. So these biomarkers or the marker sequences of the genetic element can be used not only to identify species, but may also be useful for study of diversity among the different members of the same species, for typing and subtyping of a species. So these are very important for the biologists, therefore. Now, therefore, as I said that in human genome itself, only 2 to 3% of the genome sequence is really representing the gene sequences. It is very difficult to identify which part of the genomic sequence consists of the gene sequence and which are non-coding. So this could be very easily handled by bioinformatics tools. And now a number of tools are available by which within a short period of time, given a very long genomic sequence, you can easily identify the gene sequences within the genome sequence. So let me give you a brief idea how we approach for that. It is one of the components that I'm describing, but of course there are many other features which are also relating to the gene sequence, which are also looked for by the bioinformatics tools and techniques. One important criterion or feature of a gene is to have a long open reading frame. To know about open reading frame, first we shall have to know what is the reading frame. Say, for example, this is a short sequence of nucleotide consisting of, say, around 14 nucleotides in it. Now, we do not know, it is a segment of nucleotide sequence representing a part of the whole genome. Now, we do not know whether the codon pattern starts with the first nucleotide or the second nucleotide or the third nucleotide. It may be that it starts from the first or maybe from second, the first nucleotide being the last nucleotide of the codon of the previous codon, or it may start from the third nucleotide because the first two nucleotides may be part of the codon previous to this codon. So therefore, it is not possible to understand which one is the real triplet codon representing the real gene present in the genome of the organism. Now, suppose we start from the first nucleotide, then the first triplet codon CUC will code for an amino acid leucine. Second codon will code for serine. Third codon will code for valine. Fourth will be coding for threonine. But if we consider that the coding starts from the second nucleotide, then the corresponding amino acids will vary. This time it will be serine, alanine, leucine, and proline. Similarly, if we start from the third nucleotide, then again there will be variation in the sequence of amino acid in the protein. Now, we are not sure which one is the real coding pattern which is used by the organism to code for a particular protein. How it can be, that means there can be three different reading patterns or reading frames in a particular strand of DNA. In one homologous chromosome, one pair of homologous chromosome, there are two chromatids, and therefore there are 6 different reading frames. Out of the double-stranded DNA, only one strand will contain the codes. That will be called a sans strand, and the other will be called the anti-sans strand. That will be only a complementary to the sans strand. Now, therefore, in case of eukaryotes, there will be 6 different coding frames, and in case of prokaryote, which is a haploid in nature, they will be having only 3 reading frames. Given 3 reading frames in prokaryotes even, it is not possible to easily say which one is the real reading frame, which code for the actual gene. That can be found out by one important criterion. What is that? We know from our experience that most of the functional, almost all the functional proteins in an organism are composed of around 200 amino acids arranged sequentially. For giving 200 amino acids sequentially arranged in a protein, it must have to be coded by equal number of codons. 200 amino acids is equal to 200 codons. And 200 codons equal to 600 nucleic acids. That means the nucleic acid arrangement in the actual gene will have to have contiguously presence of 200 codons with no stop codon in between. If there is a stop codon in between, that means that premature termination of the synthesis of protein will take place, and it cannot be a functional protein. To be a functional protein, it must be having at least 200 amino acids in it. So, out of the 3 different reading frames in prokaryotes, only one particular reading frame will be having an open reading frame, having contiguous presence of 200 or more functional codons without stop codon in it. So that kind of reading frames are called open reading frames. So therefore, now we have got tools which can determine and identify the open reading frames in the genomic sequence, and therefore it gives the first clue about the presence of genes in the coding sequence. There are many different gene finders and ORF finders. Of course, in addition to ORF, the regulatory sequences are also detected by the gene finders so that we can identify the promoter sequences, transcription search site, etc., which are required for expression of the gene. So these are some of the listed uh, tools which can be used for finding genes in a genome sequence. This is also another website, a particular portal, genefinding.org, which also gives you the necessary tool for detection of genes in a genomic sequence. Another important area in which bioinformatics was applied at the beginning in a larger scale, and it is one of the important areas, that is how to compare sequences, how to identify identities and similarities between sequences, and thereby finding out similarity between the taxonomic groups, or within a taxa, the similarity or identity among the members. So this is called sequence alignment. So to understand that, we have to first refer back to some terminology which are commonly used in biology. The three terms which are almost synonymously used are homology, identity, and similarity. But really speaking, in biology, these three terminologies, these terms are having little difference in meaning. Homology refers to the origin. When two sequences are derived from the same origin, that means having a common ancestry. If two sequences are having a common ancestry, they are called homologous. So homology refers to ancestral origin, and there should be the sequences should be of same origin to be referred to as homologous. And therefore, homology is a term which has got absolute meaning. There is no comparative expression. You cannot say that A is more homologous to B than C. If two sequences are homologous, they are homologous. They cannot be partially homologous, or they cannot be compared in terms of homology. Identity. Identity means complete match. If two sequences match completely, that means each of the nucleotide in each of the position of the sequence are the same. If in one there is A in the first nucleotide position, and in the second there is A to A complete identity. So that is called matching or identity. So percentage of identity refers to percentage of match. But in biology, in addition to the identity, similarity is also very important. Say, for example, among these nucleotide, as we know, there is a complementarity among nucleotides. A or adenine binds with thymine with the help of two hydrogen bonds. Cytosine binds with guanine with three hydrogen bonds. There is always an affinity of any adenine molecule in one strand of DNA to bind with the thymine molecule in the another strand by way of forming two hydrogen bonds. So A to T and C to Z, these are complementary nucleotides. So therefore, in the duplex of DNA, both the strands are complementary to each other. So these complementary sequences are not exactly identical, but they are complementary. So complementarity does not mean that they are 100% identical, but they are similar in terms of complementarity. This similarity is more, so to say, important in case of protein sequences, because the amino acids that constitute protein the 20 amino acids are usually divided according to their physicochemical structure into different groups. Say positively charged amino acid, negatively charged amino acid, acidic amino acid, basic amino acids, aliphatic amino acid, aromatic amino acid, etc., etc. So if in a particular sequence, one amino acid of a particular biochemical group, physicochemical group, is replaced by another amino acid of the same group, there may not be any impact in the phenotype because their biophysical characteristics will remain same. So therefore, mutational event which cause changes in the amino acid sequence always may not reflect any phenotype because which amino acid is replacing the amino acid in a particular sequence of protein is very, very important. If one positively charged amino acid is replaced by a negatively charged amino acid, then it is more important for its biological activity rather than when it is replaced by the amino acid of the same group. So therefore, similarity is a very important criteria for correlating the sequence of amino acid or the protein sequence to its functional activity. So therefore, we look for similarity by using different scales or scoring metrics of similarity by a process called sequence similarity search or sequence alignment. Let me give a brief example here. You see, these two sequences, suppose sequence 1 and sequence 2, we are going to compare. Arbitrarily, if we compare them just by looking for the identity among the nucleotides, starting from the first nucleotide, then initially we find that there are only few positions where there are identities among the nucleotide sequences. But when we arrange and rearrange the sequences by following certain principles of alignment, then we usually see that identity score is increasing. All these perpendicular lines represent the identical nucleotides. For doing this rearrangement, what we have done that wherever we find some dissimilarity, when there is a dissimilarity which can be reduced by putting some gap, a gap here and there, then we are using the gap there. So by using gaps here and there, we have expanded the two sequences, placed them in such a way their similarity score gets increased or their matching score is highest. So that is called the process of alignment. That means we are to do rearrangement of the nucleotide sequences by putting gaps arbitrarily or randomly here and there. And every time we see whether the matching score is increasing or not. And in the situation in which the maximum matching score is achieved, that is called as the complete alignment. So after complete alignment, we found that lot many perpendicular lines have appeared. Means there is a very high level of similarity between the two sequences. So this process I'll not explain now because there will be another extra lecture on sequence alignment. There will be also studying how the scoring is made. We can put an alignment score by using certain scoring matrices or scale of measurement, and thereby we can find out the similarity or dissimilarity between sequences. There are many online and offline tools available which can be used easily for doing this kind of alignment. Say, for example, BLAST, FASTA, Clustal W, Clustal X. These are the common tools. BLAST and FASTA are normally used to align a single sequence with multiple number of sequences present in the database. And Clustal W and Clustal X can be used for doing either pairwise alignment or multiple sequence alignment. Then, based on the variations between sequences, either identities or dissimilarities or distances, we can also correlate or compare many sequences at a time. Which one is closer to which one? Say, for example, there are three sequences, A, B, or C, whether A is more similar to B or to C. So if A is more similar to B in terms of alignment score, then we can say that evolutionary origin of A is more closer to B. That means they might have originally evolved from the same origin, or contemporarily or similar time at the evolution, they might have emerged from the same source or same origin. But C, which is having high level of variation, giving a alignment score less, that means they are distant and they are quite away in the relationship. So by this way, among a number of different taxonomic groups or different species, among species, we can establish the evolutionary relationship. And this area of bioinformatics, which is used for establishing evolutionary relationship among species by use of bioinformatics tools, is called as phylogenetics. And by using this phylogenetic tool, we can draw diagrams which are usually called as phylogenetic tree. So phylogenetic tree can be obtained by use of different tools and techniques in molecular phylogeny analysis. A number of different tools are available like phylip, pop, mega, etc. And there are a number of methods employed like maximum parsimony, distance matrix method, neighbor joining, UPGMA, then maximum likelihood, etc. And these will be taught in a separate class while dealing with the topic of sequence alignment and molecular phylogeny. Another important aspect in which the bioinformatics play a very vital role is the protein structure prediction. You may ask me why protein structure prediction is essential. Why not experimentally determine? We have got actually three different methods available for protein structure determination by experimental process. They are accurate methods. They are X-ray crystallography, NMR spectroscopy, and cryo-electron microscopy. These three methods are available to us since long back, although cryo-electron microscopy new, it was discovered in very recently in the current millennium. And for this, the Nobel Prize in Chemistry was awarded to the discoverers in the year 2017 only. But X-ray crystallography was known since 1961. And in fact, in the initial stage of bioinformatics, the bioinformatics was only dealing with the data of X-ray crystallography, because meaningful analysis and interpretation of crystallographic data to find out or determine the protein structure, three-dimensional structure of protein, was the only area which was, so to say, prevailing in bioinformatics till 1980s when the genomic era started. But NMR spectroscopy came in 1983. But although these three methods are available for determination of three-dimensional protein structure, there are a lot of limitations of the use of these methods because they are not applicable always. There are a lot many different limitations or drawbacks or demerits of this process. Let me give you one idea. X-ray crystallography, which has been most commonly used and one of the most common and useful method for determining protein structure, it requires the protein to be crystallized. Initially, it has to be crystallized first, and only in the crystal form, X-ray diffraction can be used for determining its structure. But all proteins cannot be crystallized. Even after several attempts, many proteins, particularly the membrane proteins, which are usually available admixed with lipid moiety, cannot be crystallized very easily. And therefore, our knowledge of crystal structure of protein and the final three-dimensional structure derived from X-ray crystallography is very limited. Similarly, NMR spectroscopy is also limited in its use because it also requires the that means protein should be available in solution. Only if it is available in solution, then only it can be used for NMR spectroscopy. That is also not possible. Similarly, cryo-electron microscopy is also a very cumbersome as well as, so to say, cost-intensive method, and therefore it is also not very popular so far. And also it requires high resolution structure determination and a sophisticated laboratory with high cost involvement in the infrastructure is necessary. Now we have got different databases to store the information derived out of the protein structure as well as the protein sequences. Two important resources which store the protein sequence information is SwissProt, UniProt SwissProt, and Tremble. While SwissProt usually record the manually annotated reviewed sequences, the Tremble usually store the automatically annotated and unreviewed sequences. So the sequences which are available in SwissProt are therefore more reliable because they have been already reviewed by scientists manually, and also they provide annotation also. Annotation means additional information tagged to the sequence. Similarly, if you want to see the three-dimensional structure of the protein, then you can refer back to the RCSB protein data bank or PDB. So PDB stores the information about the three-dimensional structure of proteins. Now around 180,000 different biomolecules, including proteins and carbohydrates, are, or maybe their admixture, are available. Complex admixture is also available in the PDB database. So there are three basic methods of protein structure prediction. I'll not go into detail about that. I think some other experts will be dealing with all these three methods. One is homology modeling, threading, and the third is de novo modeling. So although homology modeling is the base method, but that requires pre-existing knowledge of some sequences and the correlated three-dimensional structure, which are similar to the unknown sequence. But if you do not have any known sequence in the database for which the three-dimensional structure is already known and present in database, you cannot use homology modeling. So in that case, you have to go either for fold recognition or threading, or to the most useful method, which can start from scratch, that is called ab initio or de novo modeling, which is a algorithmic process which gives rise to the three-dimensional model of the proteins based on the physicochemical properties of the protein or electronic properties of the protein. But it's a very complex method and therefore less reliable rather than that of homology modeling or fold recognition. But even then, after refinement and use of the best technology and tools and technique, you can go up to the 99 or more percentage of refinement, and therefore that can be also completely nearing accurate to the native structure of the protein. I'll not go into this flowchart that I have shown you how to select the best method for the prediction of the protein structure. But as I already said, that there are different tools and resources available offline and online, which can be used for different methods of protein structure prediction. Say, for example, software for homology modeling are available free of cost. You can download from the websites. One is Modeler and the other is EG Modeler. Similarly, for ab initio modeling, you have online servers, portals which give you support without any cost. For example, iTusher is one of the best servers that are commonly used by researchers for modeling by ab initio process. Then after doing the prediction of the molecular model, when you predict the three-dimensional structure based on either the pre-existing models available for similar sequences, or you do it by ab initio modeling. You need to go for validation of the model structure. So for validation also, we use a method which was developed by an Indian scientist known as G.N. Ramachandran. Under his leadership, the team of workers, G.N. Ramachandran, C. Ramakrishnan, and B. Sasishekaran developed this technique in 1963. And this is called as Ramachandran plot. And this Ramachandran plot is universally used to validate or to find the accuracy or evaluate the predicted structure developed by use of bioinformatics tools. There are many different software tools available, but these are two freeware which are available in the internet. One is Rampage and the other is Project. You can use without any investment for validating or evaluating your predicted three-dimensional structure of protein. Now, bioinformatics has been used extensively in the last 2 decades in drug designing. And therefore, computer-aided drug designing, or CADD, has become a very, very important emerging area of bioinformatics. There are two approaches in drug designing. I'll be very slow, very fast, and only I'll be simply explaining some of the concepts and aspects in which bioinformatics tools and techniques can give a very important clue regarding the druggability of a molecule as well as the efficacy of a drug in terms of its biological property and its efficacy to interact with the target. So there are two approaches. One is called target-based drug designing and the other is called ligand-based drug designing. So usually drugs act by interacting with its target. In case of a infectious disease caused by microorganisms like bacteria, virus, parasites, etc., the treatment regime usually involves use of certain drugs which can kill the pathogenic organism. Okay? The killing of the pathogen is done by the drug by targeting certain receptor molecules on the surface of the cell membrane or the cell wall of the organism, which is usually inactivated or neutralized by the drug because of its very high level of affinity to the target or the receptor and its ability to neutralize it. Similarly, in case of metabolic diseases or non-infectious diseases also, the diseases occur due to certain deformity occurring in the metabolic process or the pathway of interaction between molecules in the metabolic pathway. If you can establish the so-called abnormality in the metabolic pathway and determine in which step of the metabolic pathway the deformities have occurred, we can interfere with the process by use of specific drugs or biomolecules which will either restore the normal metabolic cycle or may interfere or inhibit some of the unwanted biochemical products or enzymes which interfere in the normal metabolic cycle. So mostly the receptors or the targets are enzymes or certain protein receptors found on the cell surface of the eukaryotic organisms like human being. So either the bacterial surface proteins or viral structural proteins or the eukaryotic or human surface proteins or enzymes involved in the metabolic process are the target of interaction of a drug. Therefore, study of the drug target interaction is very, very important before deciding a particular biomolecule to be useful as a drug. So this process involves several experimental methods which are very cumbersome and time consuming. So normally in the drug discovery process, discovery of a single drug may require around 22 to 25 years prior to advent of bioinformatics tools. But now, after the use of bioinformatics tools in different steps in the process of drug discovery, the drug discovery process has become simpler and less time consuming and high cost effective. Cost is reduced by hundreds of times, and also the time required has been reduced to around 5 to 7 years. One drug can be designed and discovered in a period of 5 to 7 years against 22 to 25 years conventionally before the advent of the bioinformatics application. So either we know the complete structure of the target to explore it as the receptor molecule and then find out the biomolecules which interact very intimately with the target, or we can start with the biomolecules and based on the physicochemical structure of the biomolecule, we can also predict its biological activity and what type of target it will have highest affinity or will be capable of interaction. So all these processes can be done in the CADD or computer-aided drug designing. So we can study the drug target interaction or ligand target interaction by use of tools which are called docking tools. So docking means the study of the physical interaction between the target and the ligand or small molecule or the probable drug because of the physical similarity, because of conformational similarity or physical similarity. It is something like a lock and key relationship. A particular lock can be opened by a particular key only because they do have similarity in the ridges present in the key and the creases present within the lock. So if there is similarity between them, then only they can be opened. So similarly, if there is a conformational similarity or structural similarity between the active sites on the receptor with the probable druggable molecules or biomolecules, then there will be high level of interaction between them. So safe complementarity or configurational similarity is important here. So that is one approach. Here, of course, this reaction is static reaction. When it occurs, it is only because of conformational similarity and it is not a dynamic process. But in reality, biomolecules interact with the receptors by a dynamic process. After the physical interaction taking place, both the target active site as well as the small molecule may also undergo physical and structural changes thereafter. So this cannot be studied by the docking approach. But that can be studied by another process called molecular dynamics simulation. In molecular dynamics simulation, we apply our knowledge of statics and dynamics and the principles and procedures adopted in physics and chemistry to study the atomic interactions of molecules at the atomic level. So ultimately, we can study the dynamic interaction between the small molecule and the drug and how it undergoes changes over time. So this is a dynamic interaction. So for that also there are different platforms available. Say for example, software like Gromax, SHARM, Ember, NAMD, LAMPS, etc. are commonly employed. But Gromax being freely accessible is usually commonly applied by most of the scientists in India. So by this, you can simulate the molecular interaction taking place between the ligand and the receptor molecule by using a computer under the influence of different solvents as well as under the influence of different pressure. So that means it can be some biophysical phenomena or maybe some environmental phenomena. So you can apply that and then you can see what kind of dynamic interaction can take place between the ligand and the receptor molecule. And therefore you can study what is the dynamic interaction taking place between the two. So this is called molecular dynamic simulation. This is one of the important aspects of interaction study that is normally used extensively in drug discovery process. Another application in drug discovery process is the quantitative structure activity relationship or QSAR study in which, based on the physicochemical or electrodynamic properties of molecules, the biomolecules can be correlated to their biological activity. Means biomolecular activity of any biomolecule, a small molecule, can be predicted based on the biophysical or electrostatic properties, stereoelectronic properties. So hundreds of biophysical and stereoelectronic properties have been listed. Some of them are very important, say, for example, log p value, molar reflectivity, dipole moment, polarizability, etc. And based on similar kind of hundreds of different biophysical or stereoelectronic properties of the molecules, by use of regression equation, by using statistical method, one can predict the activity of the particular biomolecule by comparing with the standard biomolecules which are already known, and their correlated bioactivity is also known. Okay, so already hundreds, thousands of biomolecules have been studied in greater detail in terms of their biophysical and stereoelectronic properties, and their biological activities have also been determined, and they are already available in the databases. And based on the known molecules, you can develop a regression equation of this kind. Activity is equal to C plus C1 into D1 plus C2 into D2 plus C3 into D2 by using statistical regression method. And based on that, you can predict the biological activity of an unknown molecule based on the physicochemical and stereoelectronic properties. So this is called QSAR. This is also very commonly applied in ligand-based drug designing. Then similarly, you can also predict the druggability of the compound, because to be useful as a drug, the biomolecule must have to have certain specific properties. This has been listed by various scientists at different times, but the number of properties which are most important for a druggable molecule to possess have been listed by one scientist known as Christopher A. Lipinski, who developed this technique in 1997, and this is known as Lipinski's rule of 5. Of course, there are a lot many variants came up after this. So by using software tools, you can also predict the druggability. I did not refer to the software tools for QSAR in the previous slide. As I mentioned here, EasyQSAR, Pedal Descriptor, eDragon, Marvin's Cats, these are some of the tools which are used for QSAR study. Similarly, for druggability study, by use of Lipinski's rule of 5, you can use molsoft.com to study the drug likeliness as well as molecular property prediction of the drugs. Not only restricted to these properties, but we can also have computational tools, for predicting absorption, distribution, metabolism, excretion, and toxicity of the molecule by using computer. Before the actual in vitro experimentation done in laboratory, before the druggable molecule is taken to the actual wet lab experiment, the usability of the drug in terms of its absorption capacity, its distribution in the body, the metabolic process involved, and the type of excretion or the pharmacodynamic process which will be involved in total metabolism of the drug and its excretion in the body, as well as the probable toxicity with the drug, can also be studied by use of computational tools. So that kind of analysis is called ADMET analysis or ADME-TOX analysis. ADMET analysis or ADME-TOX analysis. So for that also, different online and offline tools are available. For example, ADMET Lab, M-Test Quattro's analysis tools, mobile at RBPS web portal. These are some of the important tools which can be used for ADMET analysis. Similarly, cancer genomics has progressed to a great extent in terms of the application of bioinformatics in genomic study. In comparison to other disease problems, the different kinds of cancers have been studied in greater detail, and correlating the genetic makeup of an individual with the susceptibility to cancer in terms of breast cancer, lung cancer, and different kinds of important cancer in human have been greatly established. And cancer genomics has been established as a very important emerging field of biological research. So it has also led to development of better diagnostics for identification and detection of cancers at the early stage. And also it has led to development of effective treatment strategies so that precision medicine can be prescribed for effective drug. Cancer genomics in one area which has progressed to a great extent in terms of application of bioinformatics.